Hi everyone, welcome and thank you for joining us today for our EAStalk webinar with Dr. Katerina Kousoulaki. Katarina is a senior researcher at Nofima working as a project manager focusing on fish nutrition and physiology and novel raw material processing and application in aquafeeds, novel nutritional supplements and food products. Katarina will share today with us her work and give a presentation about the microalgae applications in aquaculture, pre-processing and documentation of bioactive properties in salmon farming. Katarina has 21 years experience in project management in aquaculture and mainly marine but also plant raw material development research with PhD in fish nutrition. Currently, she is a coordinator of Aqua Bioprofit, QMAR, as well as the other large research projects, and is also participating as a work package leader in projects related to fish physiology and downstream processing of microalgae and marine biomasses, aiming at the development of food and feed products. Microalgae, primary producing organism in the bottom of the food chain, can be cultivated all over the globe in fresh or salt water, with or without light, to produce energy and essential nutrients for life. The relatively high microalgae biomass production cost for use in aquafeeds is a practical hurdle about to overcome. Appropriate pre-processing and long-term use can provide fish quality and product performances that exceeds the underlying application costs. My name is Anna Viskovic, and today I will moderate the session. EAStalk webinars, they last for 45 minutes with 30 minutes of presentation time and 15 minutes for questions and answers. When Katarina finishes her presentation, you can be unmuted and all the questions you have for her can be asked directly. During her presentation, you can send me a question request or a private message and I will put you in a queue for question and answer section. Also, I would like to ask you kindly to open now the website slido.com in another tab on your computer, tablet, or a phone. After the question and answer section, we will have time for a quick 3-question feedback from you. On each question, you will have 10 seconds to answer. We would kindly appreciate your feedback so we can work on improving our webinars for you. Later on, Katerina will share a slide with the code you need to enter on slido.com website. Many thanks, Katerina, for accepting our invitation to host an EAStalk webinar and willingness to share with our members and followers your work. If you're ready, you can start with your presentation.
Thank you very much. It is actually amazing how many have registered for this talk. Thank you all so much for being here, and also EAS for giving us the opportunity to present our research on microalgae. So there is not such a thing as a microalgae. There's not such a thing as a microalgae. So there are hundreds of thousands of different species, and they can have higher production rates and higher plants. So they also, of course, present equally many opportunities for diverse uses as feedstock. Today, I will present, not surprisingly, a salmon case of microalgae applications in aquaculture, because this is a popular product with increasing market demand over time and also political ambition to increase production. I'm quoting the Norwegian Prime Minister Erna Solberg 5 times by 2050. And it's already currently exceeding 1.2 million tons annually. So, and this growth, which is enormous and is to cover the need for high quality proteins for increased populations, so has to happen sustainable. That means both profitable and safe for the fish and the environment. So microalgae are considered to be the answer for providing at least the omega-3 long-chain polyunsaturated fatty acid in lac to support this growth. And it is also at the same time common understanding that the volumes are not there yet, and nor is the price. So for many products tested, we also see reduced nutrient availability in fish, which in the end makes this solution no sense. So to enable this growth in microalgae production, we need to work both on the technology at the same time as fish physiology to close this enormous knowledge gap. So today, I will present our efforts to this goal. I will give you a short overview of some of the projects we are working. And then I will discuss the technology side, so how we are trying to contribute to the improvement of the quality of the microalgae products, and also discussing the nutritional attributes of such products, and also briefly mention the applications in salmon production. So one of the big projects that we have been working on is the Algae to Future, where we are working with phototrophic microalgae, different species, and in 3 applications at least, which is bread, beer, and salmon feed. There are a lot of partners to these projects, and the coordinator is NIBIO, and it's funded by the Research Council of Norway, and we have both beer producer and baker and pharma companies and Norwegian and international university research institutes, algae for the bioactive producers, and a farmer which wants to use Folvengård, who wants to use the microalgae for feeds in the cows. So there's a lot of possible applications. And there is, of course, the known heterotrophic microalgae. Chytrium limacinum is the species that I've been working very much with, and this is on using a product from Alltech and funding from the Research Council of Norway, FHF, Alltech, and Salmon Group, which is a group of salmon producing here in Norway. So this species is produced heterotrophically, so in closed tanks without light. And also we have a lot of environments we collaborate with. I will mention the University of Valencia because of the results I will show you partly come from this laboratory, and the masters now graduate Maria Elena Kokali, who has been working with us, and the University of Bergen. Where do we get the algae for this phototrophic macroalgae application? So project, it is from the National Algae Pilot Plan by NOR, CO2 Bio, and located strategically in Mongstad, which is an oil distillery, with the possibility to directly utilize both excess heat and energy and the CO2 directly in the culture. And also NIBIO, coordinator of this project. So we will start with the process. And why do we need to process a macroalgae? It is, as I said, on the quality side, the reduced bioavailability of nutrients by the algae can be mostly due to the rigid cell walls that this organism has, and this has to crack open in a way for the animal to be able to use the nutrients inside. So this complex cell wall is possible to be broken open, and we are using an equipment which is called a bead mill. So why is this important at all? So the costs, which is the problem number one, for the production, let's say, and the operational production cost, and the operation and the capital cost. So this amounts of 95% of the total cost of the production of this biomass. But further, cell wall disruption, which actually will enable the availability of the valuable nutrients in the algae, it accounts for less than 0.5% of the total costs, and drying is the rest. So fish are not developed most fish are not developed to open and utilize microalgae. And this studies that is realized by the University of Wageningen, and they have shown that it is such a small amount of the total cost, the processing, and for every percentage that is not made available, saving on small processing costs, you are actually throwing a lot of money down the drain. Now you can see on your screen 2 of the studies on economics of microalgae production and processing. And you see clearly in the pictures that the whole cells on the left side is tetracell mishui, and partly disrupted and fully disrupted, which is possible with using bead milling technology. So bead milling technology is something we have chosen because it is studied and scalable and very efficient, and is happening in the absence of oxygen or heat. And it has, however, a lot of different parameters that are necessary to be optimized in order to as I mentioned before, break the cells as much as possible in order to get every single gram of it available for the fish. The way we are working when it comes to processes are to use factorial designs in order to test many parameters at the same time and receive a surface response which will show us where the optimal process parameter combination lays. And in this figure, which is just one of many we get when we are working, it shows that when you reduce the flow rate through the bead mill and you increase the dry matter of the biomass, then you improve the disruption efficiency of your biomass. So this is the way we're working. We are testing different points of different combinations, and then we get the optimal processing combination. So just a few slides on some results is presented in an easier way to grasp. So we see that when we increase also the tip speed, which is how fast the agitator inside the chamber of the bead mill, which is filled at different degrees of with beads. The beads can be of glass or zirconium, different materials, so this is something also to be optimized. Then by increasing the tip speed, we see naturally that we break more cells. And also by decreasing the flow rate, so bringing the biomass at a lower speed through the equipment, of course we break more cells. This is a chlorella. Another process that you can utilize in order to make the nutrients of the microalgae more bioavailable is pulse electric fields. This does not open the cells, but it permeabilizes them in a way that valuable components can become higher extractable. And some examples that I can give you on that is, for example, pigments. There are microalgae without treatment, are the blue, and the yellow and the orange are the amounts of either chlorella on the left side or other carotenoids extracted after treatments with pulse electric fields. Another process that is related to the production of the microalgae is the drying. Freeze drying has been tried by many or other types of drying using air and steam. Spray drying is a technology we have been working on, and it's quite efficient, and it produces a fine powder which then we can use in our experiments. And now you see many bags of different species of microalgae we have processed from Mongstad in our Aquafeed Technology Center here in Nofima in Bergen, Norway. So what happens after why do we do all that? Is this important at all? So I will show you some results of a chemical analysis of Phaeodactylum and Tetraselmis shui. When we increase the efficiency of disruption, then we increase the solubility of the protein. So that we may assume that then the protein becomes more available also for the fish. So with every small improvement we do in the processing parameters, we make more and more and more protein available from this biomass. And this was both for Phaeodactylum and also for Tetraselmis shui that we have tested. Oil is another valuable component containing also the omega-3 long-chain polyunsaturated fatty acids. We also see that we increase the extractability of oil, and then we analyze it chemically in the lab. But there are, of course, many other things present in the microalgae. It's not only protein and oil, and vitamins is one of them. They contain, many species, a lot of, for example, vitamin C and vitamin E, which are their antioxidants themselves, and at amounts like over 1,000 ppm in a dry powder. On a juice but wet, it has 500 ppm vitamin C, for example. And there are other also valuable compounds. Some examples are the polyphenols. This is Tetraselmis shui. It has high amounts of capsaicin, for example. This is present in the hot chili peppers. Maybe that's not very palatable for fish that we don't know, but it's probably something to consider for food applications. And there is a lot of cinnamic acid, or consider amounts of cinnamic acid, which also become more and more available by improving the cell wall disruption conditions. And here are listed some applications of these two phenolic compounds that we found higher represented in the two species that we have been working on. So all these compounds, they have a bioactivity. All the drugs, most of the drugs are coming from plants, and this is part of it. And we see that by disrupting more and more efficiently on the right-hand side the biomass, then the total antioxidant capacity increases of this product. And the total phenolic compounds extracted following disruption with bead milling, they were increased up to 100%. So that shows that whatever value we expect to get from the microalgae, we can't possibly get it without properly disrupting the cell walls, processing it properly. And this is the main message that I want to pass today. So we have seen that a biomass is incorporated in a diet, and with experiments and trials and publications, and if it's not, or we don't know how it is processed, then we are not sure, and we can't know that the results are to be trusted. And also for industrial applications, it is important that we always try to have a well-processed biomass for our experiments in fish physiology, for example, in aquaculture. And there is another process that comes after the previous processes I have mentioned. So that is when we want to make the fish feed, then we have the extrusion process. And with some microalgae, so we are lucky to get the cell wall disruption adequately only by this process, because it is a quite harsh process with high pressures and shear and temperature. And this was the case with the heterotrophic microalgae Schizochytrium limatinum, where we saw that by extruding alone and not other pre-processing other than drying, then the increase in the digestibility of both proteins and lipids was adequate. But that's not the case for many of the phototrophic microalgae, which has a more rigid cellulosic cell wall. So, but still there are issues on this side, because if you don't know exactly how to include the microalgae in your feed that you will produce by extrusion, then you may get surprises, as you see on the top right picture. Then there is clearly a pellet that is not optimal, and when we have analyzed the technical, physical quality of the pellets containing different amounts of Schizochytrium limatinum, which is a very oily product, then we see that we can get all kind of different types of pellets. So pellets that are not strong enough, or they are too hard, or too soft, or little or very soluble. So it is important for every new biomass that we want to include in our feed that we have also control over how to implement it in extruding and producing fish pellets. Many understand or know or haven't thought of the thought that the pellets, they have to be of a high technical quality, and that is because they have to withstand both transport and blowing hundreds and thousands of meters in tubes into the cages. At the same time, they have to be soluble enough to be digested, but not too soluble so that we lose the nutrients in the sea and in the environment. So in our study, which is published, we have seen that at least in this Schizochytrium limatinum biomass, we are able to include, this is a spray-dried powder, up to quite high amounts that are adequate to even increase the total amount of EPA in the DHA, mainly in the fish feeds. So that this work should be done also for the other species, if they should be included in high amounts in the diet. So now I have promised to show also some applications. Going first the same sequence to the phototrophic microalgae, which we have worked with together with Noor in University in Buda, and my dear colleagues Kiran Viswanath and Mette Sørensen and co. So we have tested the intact and the broken cells of Phaeodactylum and Tetraselmis. And we can see on the fat side that breaking the cells increase significantly the digestibility of the oil, both for Phaeodactylum and for Tetraselmis. On the right-hand side, we can see that this also happened for the protein in the case of Tetraselmis, but the protein digestibility of Phaeodactylum, that was actually decreased. And that is because this, what we can assume is that we release the proteins, and a lot of the proteins in microalgae are soluble and also with bioactive activity, so there can be all kinds of different nitrogenous compounds, which we want to maintain, but the extrusion will apply heat and may denaturate the proteins of the microalgae, may bind them with other components of the feed. So there is clearly something that we don't have full control over when it comes to maintaining the protein quality of the microalgae after processing and extrusion. Another result that we saw in using the processed Phaeodactylum now, so on the left the greens are non-processed, well, non-cell wall disrupted, but they are passed through the extruder, and the pink is cell wall disrupted and of course extruded. So what we see is that without adding any more pigments in the diet, the treatment with the broken Phaeodactylum tricornutum, they resulted in fish which had more strong pigmentation. This is salmon, and also the pigmentation was fortunately in the muscle, but also maybe a little bit unfortunate also in the skin. So this might be very interesting for many species. Other species maybe not so much on the salmon case. So it was possible to improve skin pigmentation by using this processed microalgae, and that is maybe due to the release of all the antioxidants, which are then protecting the color of the fish and saving the astaxanthin, which also the pigment and antioxidant, and may be used up more in the lack of other antioxidants in the diet. And this is very interesting because the previous slide I showed you was a very new results. More old results, like from last year and the years before, we have been working with Schizohytrium limatinum. We have seen exactly the same. So without adding any more pigments, astaxanthin in the diet, by using microalgae biomass, then we see increased levels of astaxanthin in the fillet. And that was over 20% of improvement in the astaxanthin content in the diet. So there is a function of the microalgae, both the phototrophic and the heterotrophic, which is of high value for aquaculture. We saw another interesting positive result using the Schizohytrium latinum in the diet, which was the reduction of the undesirable melanin discoloration spots. And this is that we don't know why it happened, but at least there was another application that may be considered when using macroalgae. And then at the same time we saw a positive increase in feed intake significantly, which resulted in fish that grew more when we fed them 6% Schizohytrium limatinum in the diet, replacing the fish oil. And this was a lifelong trial going from fresh water to salt water in cages, and that included a lot of stress and lice treatments. For the fish it was like more natural, challenging conditions for farmed fish. And then we see that the macroalgae biomass, which is an entire cell containing not only proteins and lipids, but many other components, of course, is possible to show benefits in challenging conditions. We also saw an improvement of 1 or 2% in the slaughter yield, which is, of course, another element that is of high value for fish production. Our works are very comprehensive using microalgae. We have been working on histology and gene expression with our Microarray, and we see a lot of modulation of physiological processes, all from growth, metabolism, and protein metabolism, and health, immune system, and all that is contributing to the picture. We see all the effects that are possible to obtain using this potentially sustainable source of protein, lipids, and valuable components. It is not yet there. We know that the application of microalgae is mostly in theory, in a way, and we want to see more and more coming in the use by the industry, and communication of results, the good stories that microalgae can provide is very important. I consider it very important that we go out like this webinar, and we talk about the possibilities and also the limitations and the way forward. And so we have been trying to show all our results that are coming as fast as we can. And this is the last trial, long-term trial that we have recently published. It's open access in Frontier, so we're using Schizohydrum limatinum in salmon farming, and you can visit it and see our results. And the most funny thing is that when you come out with good stories, of course everybody wants the good stories, and then they are taken up very quickly. So BioMar very quickly announced that they will use microalgae in the diet for salmon, which is a big thing, and also brought with them big salmon farmers like Leroy, and they introduced the heterotrophic microalgae in the diet, and recently also has led to another large collaboration we have, where we will use microalgae also in even higher amounts in inclusion levels in the diet, in commercial setting in 3 companies in Norway, full cycle, full production, with microalgae replacing the fish oil in the diets. So my main message today is that there are fruits and low-hanging fruits to get by using a microalgae biomass, but to get there, process is the key. So it is important to work, especially for many species, on improving the processes, and of course trying to work on the production costs to get them to a level that are affordable for the salmon farmers and the consumers. I think I was very efficient today. Thank you very much. I take questions.
Thank you, Katerina, so much for the excellent presentation. If you have a question for Katerina, now is your time to ask it. We are opening discussion for the next 15 minutes. You can send me your question, and in case you are able to talk, I can unmute you and you can directly discuss about your question with Katerina. So Katerina, we have a first question from Lady Lagos. How is the carbohydrate composition? Carbohydrates are quite important for microbiota modulation. Have you characterized it? Sorry.
Yes. In our, we have not characterized them in our studies. But of course, there are a lot of components that we assume that have contributed to the functionality of these biomasses in the gut, the fish intestine, and the absorber. We also saw improved in this last trial I mentioned with Physiology limazinum, we saw improved lipid and protein digestibility, which we didn't really expect, because we know that the microalgae often they have a lot of saturated fatty acids and also fully saturated triglycerides, which can be less digestible. But we assume that via modulation of the lipid metabolism and also transport system and the health of the gut, it was possible to actually, and where carbohydrates would have contributed, and the microbiome modulation to improve the digestibility of both proteins and lipids from this on these ingredients.
Okay, thank you. The next question is, which percentage of inclusion was tested in the in vivo trial?
Yes. In the phototrophic microalgae, we have been using 5, 8, and 15%, and the digestibility study that we tested, and it was 30%. That was the phototrophic microalgae, so it was quite a lot. And when it comes to the heterotrophic microalgae, in the published studies, we have used from 1 to 15% in the diet, and in the long-term study that we used and concluding in the cage, is then we used 6.5%.
Okay, thank you. Another question we have is from Rudrapa. Can I get the microalgae species which are used in salmon farming with their nutritional aspects?
To get the biomass or the chemical composition is published. The biomass is available by different producers.
Okay, thank you. Another one we have is from Laura. Could you please explain a little more on the microarrays figure?
Oh, it is. We have analyzed 15,000 genes. So let's say each color here would be a group of genes so it is. I can. Actually, it is also published, the results of our microalgae in a repository. So if you look for this article in Frontiers, it's open access, and it is possible to go in and find our data. So, for example, we may see genes, which is expression of genes that are involved in the immune modulation and that are differentially affected by either fish oil or microalgae, and that may be related to the very big difference in the EPA-DHA composition of this heterotrophic microalgae. They mainly contain DHA. So a lot of the results we have seen in lipid metabolism may be related to the fact that the fish compensate the lack of EPA by increasing the elongation activity of shorter chain fatty acids that are present in the diet from the plant oils into EPA, which is necessary for the immune function of the fish. Now there are a lot of we could speak forever, I think, on this part. So that was a good question.
Thank you, Katerina. Another question we have is from Mohammed. What types of algae can be used to raise colors in ornamental fish?
The colors. Of course, you would immediately think that the Hematococcus pluvialis, which is producing a lot of astaxanthin, the red pigment that we have also in salmon, would be the most that would be the obvious the choice. And there are other species that Rhodomona, so there are different species that is easy to find, and I'm glad that you contact me and I can give you lists if you find it difficult. But it is possible to find microalgae that are producing high amounts of astaxanthin and camtaxanthin, then they will give the red colors. If you're looking for green colors, then of course it's easier because chlorophyll is abundant in, or yellow lutein. So it is possible to find all different kinds of microalgae for all different kinds of colors.
Okay, thank you. Another question we have is from Tia. Have you looked at other carotenoids than astaxanthin in the fillets of fish fed these microalgae?
Yes, so astaxanthin and idoxanthin, which is a metabolite of astaxanthin, these are the two pigments that we are analyzing mainly on salmon because they are related with the uptake of astaxanthin from the diet. And there will be also other carotenoids, and we have been analyzing camtaxanthin and lutein zeaxanthin also. Mm-hmm.
Okay, thank you. Another question we have is from Maxa. Could you please tell in more detail about any contradictions?
Contradictions?
Yes, contradictions.
Is it possible for Marta to specify a little bit on the question? I don't know which contradictions.
Okay, I will, yeah. So Masha, if you have any sub-question, please let me know. Another question we have is from Christopher. Did you compare bead meal with the high pressure homogenizer on these microalgae species? Which were the results?
Mm-hmm. It's a very quick question. We didn't. So we chose one method that was also the one that was used in Algae Park and the economic studies, and we believed in this and invested on that and optimized. So we got both bad and good results with bead milling, and I assume you could get good or bad results with high pressure homogenization, and there are published research where they show that is an efficient method, high pressure homogenization as well. Our only concerns would be because of the heat involved in the high pressure homogenization may have been maybe less desirable for different compounds.
Okay, thank you. Another question we have is from Jessica. What is the % inclusion of algae with bed milling that improved flesh pigmentation and SGR?
Yeah, I think it was 8%. 8.
Okay, thank you. Another question we have is from Anna. Was microalgae biomass used to replace fish meal or fish oil?
It's fish oil from the heterotrophic, and for the phototrophic we also want to replace part of the fish meal as the protein amounts are also quite considerable. But the heterotrophic we basically replace the fish oil.
Okay, thank you. We still have a lot of questions, but I would like to tell to all the participants, in case you would like to directly talk with Katarina, just let me know so I can unmute your microphone. Another question we have is from Francesca. For how long fish were fed the microalgae feeds, and at what dietary inclusion levels to obtain such interesting pigmentation results? Thank you for the nice presentation.
Thank you for your words. And the phototrophic microalgae were used in the tank trial for 3 months. Can you hear me?
Yes, I can.
Yeah.
Can you hear me?
But also we have fish in the cages in a longer experiment. That's for the phototrophic microalgae. Again, testing broken and disrupted or non-disrupted biomass of Phaeodactylum. And that these results are not yet available for, so that would be longer than 3 months. And for the salmon study with the heterotrophic microalgae, the Schizohydrium, this study was 1.5 years.
Okay, thank you.
And I use 6.5%, so similar amount. So we are, if you want to see effects, well, at least if you want to incorporate enough biomass to claim that the effects are due to, so I think we need to promote high levels, even though they may be at the moment too costly to consider commercially.
Thank you, Katerina. And this, we have another question from Francisco. Have you found if microalgae inclusion have positive effect on immune status of fish, especially in the case of phototrophic algae?
Today I focused on what we have been doing because it's possible, of course, to go in the literature and see a lot of results from many research environments. So I have read about that, but so far this is not the focus of our studies. I'm certain that there are effects. This is the theory. In the heterotrophic microalgae, we saw mainly the growth promotion, which is not directly a health effect, and the survival was not different in the treatments. So I have no results to show you on that at the moment.
Okay, thank you. Let's see the next one. It's from Florina. Have you looked at immunostimulant compounds? Have you tested the feed on juveniles?
We tested the heterotrophic microalgae in par, so salmon in freshwater when they were small, and then we actually studied the smoltification process. The smoltification process is challenging, and so the fish go from freshwater to salt water, and it's an osmotic shock for them. And what we saw was very clear modification in the lipid metabolism. But again, in this controlled and optimal environment in the tanks and the trials, often you don't see the challenges and you don't see many effects. I will repeat that when we went out in the cages and there was a challenging. So then fish eating the microalgae, they immediately started eating more and growing more. So this is so in the total performance was improved, but the immunity of the fish, I cannot say I have direct positive results on that.
Okay, thank you, Katerina. Another question we have is from Christopher. Can you tell us more what was the protein content of your algae meal? Was it less than 60% to fully substitute fish meal?
Oh yeah, so then we go to the phototrophic macroalgae, right? And the phototrophic macroalgae in dry matter, the Phaeodactylum, that had about 45% protein in dry matter. And we have worked also with other species, but this I didn't show you. And then again, the Nanochloropsis, for example, it has about 47% protein. And then the Tetraselmis, which we also used, then that was also a little bit more. It was about 50% protein in dry matter basis.
We have another question from Christopher as well. Could you provide lipids composition of your lipid fractation of percentage of omega-3?
Okay. Then the lipid composition in dry matter of the Phaeodactylum was about 12, 15, 17%. There are different amounts, and then the Nanochloropsis is higher, so it's about 22, 23% lipids. The heterotrophic microalgae, they are higher, it's about up to 60% lipids, and then let's say in the heterotrophic microalgae can be 30% DHA, in the Phaeodactylum, or let's take the Nanochloropsis, but I didn't show results for that. But the Nanochloropsis would be about, let's say, 17–18% EPA on oil basis, and little DHA. So they have 1%, let's say, DHA. When in the case of the heterotrophic, then it's mainly only DHA and very little EPA, 0.2% in oil basis. Mm-hmm.
Okay, thank you, Katerina. The next question we have is from Anna. Considering the need for omega-3 fatty acids in the aquafeeds industry, would you consider heterotrophic microalgae more promising than the autotrophic species?
Mm-hmm. At the moment, as the situation is and the reality is, the heterotrophic macroalgae are produced in large scale, and they are in salmon feeds today. So this is not only more promising, it is actually practiced. So the question there is, will it grow even more? And in the future, when there will be needed more EPA or more DHA, will that be possible to happen from either heterotrophic microalgae, which are already in practice, or supplement with a phototrophic microalgae? Hopefully they bring with them also a lot of other components, and consider the antioxidant effects. The phototrophic, they have to protect themselves from the destructive effects of the sun. So they do develop a lot of different compounds that are beneficial for their own survival and clearly bioactive in the fish. So I would hope that, and there are a lot of activities and a lot of research and industrial work on improving the production efficiency and reducing the cost also for the phototrophic microalgae. But they are not used commercially in the large-scale big cage farming, for example, but they are used already for many years in the hatchery. So this is still, of course, valuable and important.
Thank you. We are actually past the 15 minutes, but maybe if it's okay for you to answer on 2 or 3 more questions.
Yeah, sure.
Okay, thanks. So another question we have is from Jacob. Which species did you use for the fish meal replacement trial? Phototrophic one, distributed biomass, or purified protein products?
Yeah, we used the phototrophic microalgae, the Phaeodactylum tricornutum and the Tetraselmis chewy. We had better results with the Phaeodactylum tricornutum. I don't think the fish liked very much the Tetraselmis. This is a result from Buda University. And yeah, so that was the Phaeodactylum. We didn't replace all the fish meal in the trials, only partly.
Mm-hmm. Thank you. Another one is from Christina. How does the inclusion of microalgae as a protein source impact the price of the feed as a percentage? Same for the microalgae oil, phototrophic.
I'm a researcher, and I don't even know the prices. So this is not my field. What I see is the result, and I'm sure that you would gain. Well, maybe you would have a more expensive pellet by using some algae in the diet, replacing fish oil, which will be more inexpensive. But if we have growth performance improvements or pigmentation improvement, then I'm sure it will be worth it. So you will make money in the end. When it comes to the phototrophic microalgae, the literature shows they are many times more expensive. So that would both make the production more expensive and the feeds, of course, more expensive, and is yet to be proven that you will make your money back. But there is a good chance you may do that.
Okay, thank you. Another one is from just now. What species have you worked on before? Only on salmon, or?
No, we started working with the Schizohydrium limatinum from 2012, and only worked with these species when it comes to heterotrophic microalgae, and it was the Alltech that was the producer. And I have not worked with any other heterotrophic microalgae in salmon production. In the old times, we came across spirulina or other species, and then you can get also nice green pigmentation in your fish. And now we are working with all these different phototrophic microalgae, which I mentioned was the Nanochloropsis and the Tetraselmis and the Pheodactylum, and I have not worked. But this is, I deal with this webinar as a conference presentation when I show my results and the background, and the other results are in the available online.
Yes, thank you. This will be actually the last question for today. It's a question from Ratna. For the algae which causes melanosis in flesh, is it also affect to the fish health? And I wonder if the application of this algae would have the same results on pigmentation in other species.
Yeah, so that was what we saw is by increasing the microalgae in the diet and replacing fish oil. So what then we saw in this treatment less or almost no melanin spots. So it decreased the amount of melanin spots in the fillet, which is a very positive effect, which was not the hypothesis or expected, and this is of enormous economical value, because then you don't degrade your fillets and you maintain high prices and value of your product. And it will not, I assume that will not be directly an indication of, it will not be a direct health improving effect, but if fish are able to have, let's say, less cellular stress and produce less melanin, then you may assume that they all are healthier also. So they have a better oxidative status. That's an assumption.
Okay, thank you, Katerina. We still have a lot of questions, but I think we are past 8 minutes past the time we should be done with it. So in case you have here the Katarina's email, so if you have any other question for Katarina, feel free to ask her or to send me an email and I will ask directly Katarina. As I mentioned before on the beginning of the webinar, can you please go to slido.com website and type the code 2605. We have a quick feedback from you, so if you can please answer on the 3 questions we have. Also, on each question, you will have 10 seconds time to answer it. We received, Katarina, really nice feedback from the participants, and they are thanking you for a really good presentation.
Thank you also for being here. It's incredible and also very strange.
Strange to have a webinar? Or
not to see any of the participants. But that's a new reality.
Yes, that's the new way of...
We keep the wheels spinning. We have to.
Yes, yeah. And also for the people that maybe had a problems with the sound or they sign in late, the webinar is recorded. So in a few days you will get a video of the webinar. So don't
despair.
Yes, yes. Let's wait. I will wait 5 more seconds because for the first question, it was necessary to have a bit more time for people to log in to slido.com. But I will now switch to the other question. So please feel free to answer on it. Also, in the meantime, when you answer the question, I would like to thank you all for participating in the webinar. And our next webinar will be on June 16th with Joanna Gosling and James Taylor. They will discuss more about the maximizing the potential of coordinated national research for the Arch UK. Thank you so much. So let's see. Yeah, Katarina, thank you so much. Thank you for a nice presentation.
Thank
you.
You.
Really nice, really nice feedback.
Thank you.
And this is the third question I have for you, and also the last one. Great presentation. So I would like to wrap up now, and thank you once again, Katarina, for accepting our invitation to have the webinar. The feedback was really nice, and yeah, and that says all about your presentation and your knowledge in this field. So thank you all for participating. And in case you would like to suggest or propose any topic or presenter, please send me an email on anna@aquaeas.eu. Thank you so much once again.
Yeah, thank you.
Thank
Bye.
you. Bye.